Hello students, welcome to Aakash. I'm your botany faculty, Dr. Sukirti, and today we are here to discuss the paper that is mock test 5 of AIATS Plus 2025 phase 2, which was held on 16th of Feb 2025, right? So why we are discussing this particular paper, because lots of you have given this particular paper and done few mistakes, and to analyze those and to know about those gray areas, not only how to deal with the question but also the entire periphery of that topic, that's what we are going to discuss over here. And the chapters that we are dealing with in this particular lecture would be principles of inheritance, cell cycle, ecosystem, respiration and microbes, right? So firstly, we'll begin with the first question. So very good morning to all of you. So the first question over here says that given below is a pedigree showing inheritance of myotonic dystrophy, and the dark ones are affected individuals, right? So when we talk about this myotonic dystrophy, it is basically a genetic disorder in which the muscle wasting is going to be there. And when we talk about this myotonic dystrophy, it is an autosomal recessive disorder. Sorry, really sorry, it's not recessive, rather it's an autosomal dominant disorder. Right? So when we know about this particular fact that this is an autosomal dominant disorder, so with this pedigree we could simply assume that all the unaffected individuals must have been homozygous recessive, right? Because they are lacking the dominant allele, and this is because of the fact they are unaffected, right? So in this particular pedigree, as it has already been mentioned that the dark ones over here are the affected individuals, that means all these ones, right, which are non-filled symbols, circles and the squares, they are going to be the unaffected individuals, and all of them are going to show, let's say we are assuming the allele to be A, so all of them will be having the genotype small a, right? Now how we are going to define or decipher the genotypes of these individuals? So what we know over here that this particular female, as it is being homozygous recessive for this particular trait, so it must have received one allele from the father, that's small a, and one from the mother, right? Because we know that the individual get one pair from one parent and another pair from another parent, right? So over here one allele must have come from here. So this particular female, the affected one, is going to be heterozygous for the trait, right? So you should know that how you are going to analyze the pedigree so that you could decipher all the possible consequences, because certain times you are not going to be asked for that what kind of genotype is being shown. Certain times they will ask you about the inferences as well, right? Now going by this, as this particular unaffected individual is having small a allele, by the same thing, one allele must have been from the mother and another from the father, as this is an autosomal one. So as the father is affected, the other one, the other allele that it will be having would be capital A. So he is also going to be homozygous. Now as if you perform this particular cross, right, capital A with small a, you can have two types of genotypes possible. One is capital A and another is small a. Small a will be the unaffected individuals, right? They are going to show this particular genotype, and the affected ones are going to exhibit this genotype. So all of them over here in this generation would be heterozygous for the trait, right? So this is how we could decipher the entire genotypes for the individuals that are being depicted over here in this pedigree. Now the question is asking about the possible genotype of person A, and this is the person A, as it is unaffected and the disorder is autosomal dominant. So the genotype it's going to exhibit will be small a, homozygous recessive. But because over here in the myotonic dystrophy they have took the allele M, right? So this genotype will be considered as small m. That's correctly depicted in option 4, right? So this is how you will be solving this pedigree. Now, let's analyze this pedigree on another angle as well. Like this particular pedigree is true for autosomal dominant, right? Sometimes the question comes in a way that you have to identify that whether it is an X-linked, dominant disorder or a recessive one, or an autosomal dominant and autosomal recessive one. So we have given in this particular one that this is autosomal dominant. Now let's analyze this same pedigree, that whether it fits into any other form of inheritance pattern or not. So let's consider that the disorder that's in question is an autosomal recessive one. So let's analyze that whether this pedigree fits into an autosomal recessive pattern or not. So if we are considering that the disorder is going to be autosomal recessive, let's say, right? This time it's changed. Similar sort of question if been asked, and they are giving you an autosomal recessive trait, like if they are saying that the person is inflicted with sickle cell anemia or maybe thalassemia. In that particular case, all the affected individuals are going to be homozygous recessive, unlike the autosomal dominant one where the unaffected ones are homozygous recessive. So all of these, the affected ones, are going to show homozygous recessive genotype, like this, right? Now, on the similar grounds, as this particular individual is having two small a allele, one will be from the mother, another will be from the father, right? And as the father is normal, the other allele that it must be having would be capital A. Because of this dominant allele, the small a allele which can cause the disease is suppressed now, right? Now, similar way, over here, this particular parent will also be heterozygous because this individual must have received one small a from the father and one from the mother, right? So, as the mother is normal, he'll be having another allele as the dominant ones. And here again, only one possibility is there, like you can have two type of genotypes, one is capital A small a and small a small a. These ones will be affected, only one genotype possibility of the unaffected individual. So now this will become capital A small a, right? So this pedigree is also suited for the autosomal recessive disorder, right? That's why they have specified here clearly that if you're supposing that the pedigree is true for myotonic dystrophy, right? So in this particular case, if this question would have said that the given below is an inheritance of sickle cell anemia, and then what is the genotype of A individual, then you would have said capital M, small m, right? Now, let's consider the possibility, let's check that whether this particular pedigree is true for X-linked recessive one. Right? So, if this particular pedigree is true for X-linked recessive ones, then the females will have both the recessive, alleles for it. So, let's suppose this individual, this female is affected, so she will be having the diseased homozygous combination and the males are going to be, because they have only one X chromosome, they are going to possess that particular allele over there. So, this will be XAY, this will be XA, and it will be XAY, right? Now, for the disease to be X-linked recessive, this female must have received one X from the father, and of course the father is diseased, and one from the mother. But because mother is normal, so the other one, the other allele that's present on the X chromosome would have been dominant. So till now it is satisfying the pedigree. Now coming over here. Now this particular female must have got one XA from the mother and another from the father, right? And like males have only one X chromosome. So if this particular allele have came from the father should have been affected. And as over here you can see that he is unaffected, that is why we can infer that this pedigree does not suit the inheritance pattern of X-linked recessive disorder, right? So for that, it's not true. Now, similar grounds, let's consider that if this particular one is true for X-linked dominant, let's consider this scenario now, that whether it can suit X-linked dominant inheritance pattern or not. So if we are considering it's an X-linked dominant disorder, then the females would have been, the unaffected ones would have been showing the homozygous recessive genotype. So this particular female is going to be like this, XX, right? X would be having the recessive one, and A would be the one that is going to carry the disease. So this particular individual is going to be like this also going to be like this, right? Now, on the similar grounds, if this female is having two X chromosome, one would be from the father, one would be from the mother. And because mother is affected, so she must be carrying the disease causing allele, the dominant one, right? Then coming to this particular part, so it must have received, this particular female received one X, you know, like let's suppose we are talking about this individual XY now. So Y will be from the father and X will be from the mother. This is being satisfied. Now this particular individual must have received XA from the father and X from the mother. So this particular pedigree suits the, what you could say, the inheritance pattern of X-linked dominant disorder also, right? So this is how, if the disorder is not being specified in the question, then you have to look for that whether this particular pedigree is satisfying some other inheritance patterns or not, right? So this is a way to deal with the pedigree questions. I hope this is clear to everyone, right? And now let's move ahead to the next question. So the next question, again from the principles of inheritance, it's a very important chapter with respect to the NEET examination. A lot many questions have been came from here, and molecular basis of inheritance, right? So over here this question talks about that the gene concerned with beta-thalassemia in human being is present on chromosome, right? So when we talk about this disorder, thalassemia, firstly, it is a quantitative disorder. Right? Why we are calling it as a quantitative disorder? Because the severity of this particular disease is based on the fact that how much less synthesis of the globin chains of hemoglobin is occurring, right? So hemoglobin is composed of various chains like alpha chains, beta chains, right? So if there will be a reduced synthesis of either of the chains or both of them, that's how we are going to decipher that what kind of thalassemia the person is having, right? Let's suppose there will be a reduced synthesis of alpha chains, then we are going to call it as alpha thalassemia. If it will be a reduced synthesis of beta chain, we are going to call it as beta thalassemia. And in case it will be reduced synthesis of both of them, we are going to call it as an alpha beta thalassemia, a rare form of this disorder. Also, the amount of reduction is going to decide that whether it's a minor thalassemia or a major one, right? If let's say there will be a reduced synthesis by 20% or 25%, it will be minor. Greater than 50% would be the major one, right? So this is how we decipher about this disorder. Now here the question explicitly asking about beta thalassemia. That means there will be reduced synthesis of beta chains, right? So we often, compare, like the NEET questions comes up in this way, that they often compare this disorder with sickle cell anemia, right, which on the contrary to thalassemia is a qualitative disorder, right. Over there, quantity of globin chain is not at all affected. Rather, there will be a synthesis of an incorrectly functioning globin molecule. Over there also, beta chain is being affected. Here, beta thalassemia, reduced synthesis, there will be a incorrectly functioning beta globin chain, right. So Purvi is saying that myotonic dystrophy is autosomal recessive. No, myotonic dystrophy is autosomal dominant. There are certain forms, right? A muscular dystrophy is a very big term which is basically talking about the muscle wasting, right? But there will be variants of it, and few of the variants could be autosomal recessive, but because we are talking in terms in the periphery of NEET, and here the muscular dystrophy that we are dealing with is autosomal dominant disorder, right? That's been explicitly being mentioned. So usually myotonic dystrophy and an autosomal dominant disorder, right? I hope this is clear, Purvi. Okay. So moving ahead, when we talking about the beta-thalassemia, so there will be reduced synthesis and it is controlled by a single gene, that is HBB. Because of certain mutation in it, there will be a reduced synthesis of beta chains. And this particular gene is present on chromosome number 11, right? On the contrary to this, if we talk about alpha thalassemia, then it is being controlled by two genes, that is HbA1 and HbA2. And these two are present on chromosome 16, right? So this is what thalassemia is all about. And now because the question is asking that the gene that is concerned with beta thalassemia, that means they are talking about the HBB gene. So that is being present on chromosome. So that's present on chromosome number 11, correctly depicted in option number 4, right? If we talk about option number 1, 18, that's not true. Now why 18 chromosome is like important to know? Because the chromosomal disorder, this is a Mendelian disorder where a gene is affected. Now there are chromosomal disorders as well in which there will be an anomaly in their number, right? Or maybe there will be a truncation of the chromosomes, or there will be an addition of it, duplication. So certain how the chromosomal structure or the number is the thing that's going to be impaired during the chromosomal disorder. So over here, the trisomy of chromosome number 18 is being considered as Edwards syndrome, right? This question has been asked in the PYQ as well. Now, talking about option number 2, 16, as I've told you that the genes that are controlling alpha thalassemia, the synthesis of alpha chains are being present on chromosome number 16. And then when we talk about chromosome number 21, so the trisomy of chromosome number 21 is being considered as Down syndrome, right? So this is why you should remember these chromosome numbers as well, because all the time the question won't be coming from the thalassemia part. You may be asked the questions from the chromosomal disorders as well, right? Yes, that's correct. 21 chromosome is related to Down syndrome, right? So its trisomy is going to be considered as Down syndrome. So this is how you'll be solving this particular question. Now moving ahead, we have another question that talks about that we have to choose an incorrect option for sickle cell anemia, right? So as I have told you that sickle cell anemia often get compared with thalassemia because the two disorders, the previous one that we have discussed is a quantitative one. Over here that we are discussing is a qualitative disorder, right, in which there will be a mutation, a substitution mutation that occurs at the 6th position of the beta globin chain of hemoglobin, right? And because of that particular mutation, right, glutamic acid is going to get replaced by valine, right? And because of that particular thing, the biconcave shape of RBC is going to change itself to a sickle shaped RBC, which is having low oxygen carrying capacity, right? So as we have to talk about the sickle cell anemia, the statement that's not going to true for sickle cell anemia. The first one over here says that individual with heterozygous genotype will be unaffected but will be the carrier of the disease, right? In order to know about this, we should know that what kind of inheritance pattern is being shown by sickle cell anemia. So this disorder inherit itself in an autosomal recessive fashion, right? So if it is autosomal recessive fashion, that means all the affected individuals are going to be homozygous recessive for the trait. So it is saying that individual with heterozygous genotype will be unaffected but will be the carrier of the disease. So this is true. If the individual is going to be heterozygous, that means one dominant allele is going to suppress the recessive allele that is causing this particular disorder. So this individual will remain unaffected, but it will be the carrier of the disease because it is still carrying the disease, the disease carrying allele over here, right? So this is a correct statement. Then the second statement says that it is caused due to substitution at the 6th codon of beta globin chain from GUG to GAG, right? So that's also true. Then it is said that this disease can be transmitted to offspring if both the parents are heterozygous for the gene. Again, this is true. Why? Because if both of them are carrying the allele, the disease carrying allele, so of course there will be a possibility that the progeny can have small a, the disease carrying allele in homozygous form, and that's how they can have the affected individual. Fourth one is saying that the mutant hemoglobin molecule undergoes polymerization under low oxygen tension, right? This is also true, and because of the polymerization under low oxygen tension, the hemoglobin molecule changes shape. So coming back to option number 2, right? So the mutation does occur at the 6th position, but how this particular statement is incorrect? Because this is the opposite that happens, and because of this glutamic acid is going to be replaced by valine, right? So this is the incorrect one, so that's why correct option for the question, right? So I hope this is clear to everyone that why it is incorrect, and all of these statements are correct with reference to sickle cell anemia, right? Okay, moving ahead, we have another question which talks about that which one is not an autosomal recessive disorder, right? So Sumit, this is the 12th topics that are being discussed, but this particular paper is having the entire syllabus covered, but in this session we are covering few chapters like principles, cell cycle, respiration, microbes and ecosystem. So few topics from class 12th and few from class 11th, right? Alright, so this is talking about that which one of them is not an autosomal recessive disorder. So among them, if we talk about thalassemia, as we have already discussed, that thalassemia is an autosomal recessive disorder in which there will be reduced synthesis of the chains of hemoglobin molecule, right? So this is an autosomal recessive trait. Then talking about sickle cell anemia, again that we have discussed that sickle cell anemia, over here there will be a substitution mutation that is taking place, and it's a qualitative disorder in which there will be substitution mutation that results into the synthesis of an incorrectly functioning globin chain, right? Then we are talking about color blindness, right? So over here this is an X-linked recessive disorder, right, in which the color perceptive components of your eyes are going to get affected. So this is an X-linked recessive disorder. Then we talk about fourth one, that is phenylketonuria, which is an inborn error of metabolism, right, in which an enzyme which is known as phenylalanine hydroxylase, which basically converts phenylalanine into tyrosine, that's going to get affected, right, phenylalanine hydroxylase. This particular enzyme is not being found in the affected individuals, due to which this conversion is not taking place. And now if this conversion is not taking place, what's going to accumulate? Phenylalanine, right? As it's not a stable form, so this is going to convert into phenylpyruvic acid and certain other derivatives because of the metabolic reactions. But the accumulation of these in the brain region is going to cause the mental dysfunction, right? So mental retardation is one of the prime features of phenylketonuria, which is an inborn error of metabolism, and it also inherits in autosomal recessive fashion, right? So yes, red and green cones that are going to affect it in color blindness. So the question is explicitly asking not an autosomal recessive trait, correctly depicted in option number 3, right? There is one more X-linked recessive disorder that's been, mentioned in NCERT, that's hemophilia, right, in which the clotting factors get affected. So you are not going to get the disorders like this also, you may get them as the pedigree questions also. So you should know the inheritance patterns of certain diseases, right? If they are autosomal recessive, disorders, then all the affected ones are going to be homozygous recessive. If they are dominant disorders, then the unaffected ones are going to be homozygous recessive for the trait, right? This is how you'll be solving the questions related to disorders. Now let's move ahead. Another question, and this question talks about the gene responsible for the disorder phenylketonuria. As we have discussed in the previous question only, that this particular disorder inherits itself in an autosomal recessive fashion, right? And this particular disorder is an inborn error of metabolism where an enzyme known as phenylalanine hydroxylase is not being present in the affected individual, due to which the conversion of phenylalanine to tyrosine is also not taking place, right? So the first thing this question says that the gene responsible for this disorder is a pleiotropic gene. So this is true, right? Now what is a pleiotropic gene? So usually what we see till the Mendelian genetics was into picture, that a single gene affects a single character, right? One gene, one phenotype effect. Later on we have found out that it's not always that a single gene is going to carry or exhibit a single phenotype. Certain times a single gene is controlling a lot many characters, right? So if a single gene is controlling many phenotypic effects, that particular gene is considered as a pleiotropic gene. And the gene that controls phenylketonuria is definitely a pleiotropic gene, because apart from having the effect on the mental impairment or mental health, at the same time it also controls the skin pigmentation or hair pigmentation as well, right? So that's why it is controlling multiple effects, a single gene. That's why we are considering it as a pleiotropic gene. Now B says it is an autosomal recessive gene. As I've told you that it inherits itself in an autosomal recessive fashion. That means that if the gene is present in its homozygous form, then only the individual is going to get affected by the disorder. So this is true. Then third thing is saying it is a codominant gene. Now what's a codominant gene, right? Again, till the Mendelian genetics is in the picture, and if we consider the law of dominance to be true, so according to that particular rule, what happens, what we consider is that for every character you will be having two alleles, in which one of them is going to be dominant and other one is going to be recessive. The dominant expresses itself, right, either it's in homozygous condition or heterozygous. The recessive will only get its expression done if the dominant allele is absent, right? So for everything you will be having two alleles. Now what does a codominant thing says? It's an exception to the classical Mendelian genetics dominance, because the codominant one says that there can be more than one dominant allele. Like if we see the case of blood group, human ABO blood group system, there are three alleles, IA, IB and small I, right? And over here the dominance of IA and IB is equal, but both of them are dominant over small I. So what does this mean? It means that in case the genotype appears to be IAIB, carrying both these dominant alleles, so in this particular case both of them will express itself, right? So the phenotype we are going to get will be the AB blood group, right? Unlike the law of dominance says that there will be only two, and one of them is going to express itself in all the conditions, right? So when we talk about the phenylketonuria one, it is showing autosomal recessive trait. It's not a codominant one. There is a dominant allele, there is a recessive allele, and phenylketonuria controlling gene, if it is there, then it will be the recessive one that's in the picture. Now D says unable to synthesize an enzyme which converts phenylalanine into tyrosine. As we have already discussed, this phenylalanine hydroxylase is not being found in the affected individual, right? So this is the only incorrect one, C part. So over here we have to identify the correct one. So it is saying only B and C are correct, but that's incorrect because A is also true. Then it says B and D are correct, right? Oh, sorry. Over here they are saying C is correct, but it's incorrect over here. That's why we have ruled out this option. Over here they are considering only B and D to be correct, not A. That's why incorrect. A, B and D are correct. This is the correct option, and only D is correct. Again, incorrect one. So over here, because of the correct features depicted in A, B and D, the correct answer for this question is going to be option number 3, right? So this is how you'll be solving this question. I hope this is clear to everyone. In case you have any doubts, please comment on the chat box, right? All right. So moving ahead, we have another question, and now this time it's a topic from 11th. That's cell cycle, and it says meiosis produces, right? So when we talk about meiosis, so we know certain features like this is a reductional division, right, in which whatever ploidy the mother cell is having is going to get reduced to half in the daughter progenies, right? So over here it says meiosis produces 4 genetically similar daughter cells, right? So till here when it is saying that it will be 4 daughter cells, that is true. Why? Because when it starts up, then after meiosis 1 you will be having 2 daughter cells, and then further after meiosis 2 you will be having 4 daughter cells, right? But a characteristic event of meiosis is crossing over or recombination, right? And because of that recombination, there will be genetic difference. So there will be the production of genetically dissimilar daughter individuals, right? So that's why it's an incorrect one. The next option is saying diploid daughter cells always. So that's incorrect again, because as I've told you, it's a reductional division, so it's going to reduce the ploidy. Then 2 genetically similar daughter cells, again incorrect, there will be the production of 4. And then 4 genetically dissimilar daughter cells, as many of you are saying, right, Purvi and many others, that it is going to be 4th. So that's correct, there will be production of genetically dissimilar daughter cells, because in meiosis 1 they have already been gone through recombination, where there will be a genetic exchange of the segments between the non-sister chromatids of homologous chromosome, yielding new genetic combination, and that's really important for the evolutionary processes as well, right? So this is how the correct answer for this particular question is going to be option number 4. Now let's move ahead to another question, again dealing with cell cycle, and it says that dyad of cells produced in reductional division, right? So as they have mentioned over here, we are talking about, so there is a question coming out that can you able to explain tetrad or bivalence with respect to homologous chromosome? Okay, so when we talk about homologous chromosomes, right, so there will be a pair sort of a thing, sorry, like this, X-shaped chromosomes, right? So this pair of homologous chromosome, if they are alone, we are going to call it as a pair of homologous chromosome. But during zygotene, when the synaptonemal complex gets synthesized and this ladder-like, complex joins these together, right? This process is known as synapsis. All of you must have heard about it. So when they become together, now they are working as a single entity, right? This single entity, because it carries 2 chromosomes together, we're going to call it as bivalent, right? And if we count the number of chromatids over here, there will be 1, 2, 3 and 4, because 4 chromatids are going to be present. The same structure could be called tetrad as well, right? So this is tetrad, we call it in terms of chromatid, bivalent in terms of how many chromosomes, because now this is a single entity, not 2 different chromosomes, right? I hope this is clear. All right. So the next one over here says reductional division, that's meiosis. Then dyad of cells. Dyad of cells means there will be the formation of two cells, right? So what we see, as I've told you, that during meiosis, when the meiosis 1 is completed entirely, we'll be having two daughter cells having reduced ploidy already. So these two chromosomes will be considered as dyad of cells, right? The name itself suggests dyad means two. So this is dyad of cells. So they are saying that a dyad of cells produced in reductional division, first it is saying that it is formed just after diakinesis, right? So we could rule out this option. Now why we have ruled out? Because when we talk about diakinesis, it is the last substage of prophase 1, right? In prophase 1, what we are trying to achieve is, firstly during leptotene, the beginning of condensation, during zygotene, the synapsis process where the homologous chromosome are going to get paired, during pecitine, the crossing over, the main key event of meiosis, then during diplotene, the dissolution of synaptonemal complex, and further during diakinesis, terminalization of chiasmata, and along with that certain other important events of prophase like the formation of asters in animal cell, or disintegration of nuclear membrane and nucleolus, right? So when the prophase is completed, after that also there are certain steps of karyokinesis that remains, like metaphase, where there will be an alignment of those chromosomes on the equatorial plate. Then further in anaphase 1, there will be separation of homologous chromosomes, and further in the telophase, there will be again reassembly of the nuclear membrane and the nucleolus. So when the karyokinesis is completed, after that cytokinesis has to take place, right? Because one cell division completes only when karyokinesis is completed, and then further cytokinesis is completed. Only then you will be having two daughter cells with two nuclei, and you will be forming dyad of cells. This option is saying it is going to form just after diakinesis, just after prophase. Karyokinesis is not still completed. How you are supposed to have cytokinesis? So that's why we have ruled out this particular option. The second one is saying have haploid nuclei. So this is true. It is because there is a separation of homologous chromosomes over here that reduces the ploidy to already half. So the mother cell is going to be diploid, but the daughter cells that you are going to have after meiosis 1 will be having haploid number of chromosomes, right? So this is correct, as being told by many students that it's going to be 2. Then third one is saying have bivalent chromosomes, right? This is also incorrect. As I've told you, bivalent chromosome means paired together. Over here separation has already taken place, so they are no longer in the bivalent form. So that is why we are going to rule out this option. Then fourth one is saying undergo crossing over. That's incorrect. Dyad of cells are being produced after cytokinesis. This event, crossing over, where the exchange of genetic material is taking place, has already been done during prophase, and the substage in which it is occurring is pecitine. So over there it has been done a long ago, right? So that's why this particular option is also incorrect. So with respect to the question, the dyad of cells produced in reductional division, it is going to have the haploid nuclei, right? So this is the correct answer, option number 2, right? Okay, moving ahead, we have another question which talks about again cell cycle and says that the last stage of meiosis II is characterized by, right? So meiosis II, the second phase of the reductional division. So after meiosis 1 is completed, the cells will undergo meiosis II, and for meiosis II, one popular line that we phrase constantly is that it is similar to mitosis. Because why we are saying it? Because over here there will be no further reduction in the number of chromosome. The chromosome number is going to remain the same with respect to the daughter cells that you have produced after meiosis 1. So when we say that the last stage of meiosis II, so whenever we depict this thing, that means we are talking about the last stage of meiosis II, the karyokinesis last stage is going to be telophase, right? So over here it says that formation of cell walls around the nuclei. So this is incorrect, right? Cell wall formation is not going to occur around the nuclei, but if it's a plant cell, then it will be around the cell membrane, right? So that's why this is an incorrect option. It says separation and movement of chromosomes. This is also incorrect, because when we talk about separation and movement of chromosome, this has been occurred during anaphase 1, right? Anaphase 1, you have the separation of homologous chromosome, and why we are saying movement, because they are moving towards the opposite pole. So this is an event of anaphase and not the telophase. Now third one is saying disintegration of centrosomes, right? This is disintegration of centrosomes, right? So this is correct. Last stage of meiosis II, if we talk about, this is telophase. Over there, centrosomes are not required, anymore. And then we have enclosing of two group of chromosome by separate nuclear envelopes. So there will be not two groups of chromosome, it will be a single group of chromosome that's going to be available, right? So the correct answer is going to be option number 3, right? Moving ahead, we have another question. That's question number 108, which talks about conversion of 2-PGA into PEP during EMP pathway also leads to release of, right? So this is a question from respiration part, which talks about that the 2-PGA is converting into PEP during EMP pathway leads to the release of. So when we say the EMP pathway, that means we are talking about glycolysis, right? So when we talk about glycolysis, over here this conversion step 2-PGA into PEP, it is important, and why it is important, because PEP further is going to create pyruvate molecule, right? And when PEP is creating the pyruvate molecule, that will be substrate level phosphorylation reaction, and this conversion 2-PGA to PEP is releasing the water molecule, right? Because this particular reaction 2-PGA to PEP is catalyzed by enolase enzyme, right? And this enzyme is going to release a water molecule from this, so that this PGA could convert into phosphoenol pyruvate. So option 1 over here says that it leads to the release of ATP by substrate level phosphorylation. This is incorrect. ATP release is going to take place when PEP further gets converted to pyruvate, right? And we are going to call this particular reaction a substrate level phosphorylation reaction. Why? Because over here the substrate PEP is going to donate its electron to ADP so that ATP can be formed. So over here a substrate is directly donating the phosphate to the ADP molecule for the production of ATP. That is the reason why we call this particular reaction as substrate level phosphorylation reaction, right? But that's the next reaction, not this one. The question is being demanding about this particular equation. So this is an incorrect option. Option 2 says also leads to the release of redox equivalence. So when we talk about the redox equivalence, that is in the form of hydrogen ions, and this particular release occurs when 1,3-bi-PGA converts into 3-PGA, right? Sorry, not this reaction. When basically G3P, glyceraldehyde 3-phosphate, converts to 1,3-bi-PGA. In this particular reaction, the redox equivalents are released and the NAD plus gets oxidized into NADH plus H plus, right? So during this particular reaction, there will be release of redox equivalents. Then the third one says release of carbon dioxide. So during the entire process of EMP pathway, there is no release of carbon dioxide at all, right? So that's why this is also incorrect. And option 4 says water. So yes, enolase enzyme helps in the release of water molecule during this particular reaction, right? So the correct answer is going to be option number 4, right? 105, okay, sorry. Okay, let's get back to the one that you have said, 105. Oh, okay. Oh yes, you are right. So over here the answer is going to be option number 4. I have incorrectly explained it to option number 3, right? Because this is talking about the last stage of meiosis II, which is going to be telophase. Over there, disintegration of centrosome is not going to take place. Why? Because when we talk about centrosome, it is a structure made up of centriole. It's already going to be present, you know, during the cell. Jitna bhi time the cell is going to have, they are going to have the centrosomes in it. So that is why there will be no disintegration. There will be disintegration of mitotic apparatus, not the centrosome as such, because this is an organelle. And option 4, when says enclosing 2 groups of chromosome by separate nuclear envelopes, that is correct, because the reintegration of, you know, nuclear membrane and nucleolus is going to take place. So that's why this particular one is the correct answer. So I'm really sorry for the previous one. This is the option number 4 which is going to be correct, right? Now, I hope this is clear to everybody. So getting back to the next one, over here the option is going to be option number 4, right? Is it clear to everybody? In case you have any doubts, please put on the chat box. And really sorry about the last one. Now moving ahead, we have another question, which is talking about which gases are produced in anaerobic sludge digesters during sewage treatment, a question from microbes, right? So it talks about the anaerobic sludge digester, right? So when we talk about anaerobic sludge digesters during the process of sewage treatment, so this is a component of secondary treatment, which is a biological treatment in which we use the microbes, right, for the treatment of sewage, right. So when we talk about the anaerobic sludge digester, so what happens during the process of sewage treatment, that initially we go for the physical separation of, let's say, the debris or the grit, right. We remove that particular part, the physical impurities in the sewage. Then we proceed it for the clearing of the organic matter that is polluting that water. So after the primary effluent comes in for the secondary treatment, it firstly goes to aeration tank, right. During this particular, you know, treatment, the treatment that's occurred during the aeration tank, there will be aerobic microbes, right, which are being considered as flocs, the structures which are having aerobic bacteria along with fungi. They are going to digest the aerobic, the organic material that's being found in the sewage. Then further, it goes to settling tank to remove that debris, and finally it goes to an aerobic sludge digester, right. Over here the anaerobic microbes are going to perform the digestion further, and over here a major section is of methanogens, right. So during this anaerobic sludge digester thing, the production of biogas, you know, or you could say methane is going to be there. Methane, of course, in the large quantity, but other gases as well which are being produced as a byproduct by methanogens. So that is why you also, you know, read this particular line that the, sludge, anaerobic sludge can go directly into the biogas plant for the production of methanogens, right? For the production of methane, not methanogens. Methanogens are going to produce it. So as there will be a production of biogas, so the components would be methane, the highest. Then there will be production of hydrogen sulfide also, a bit of carbon dioxide also, right? So the first option is saying methane and carbon dioxide only. So that is incorrect because you may found the production of hydrogen sulfide as well, which is also one of the prominent contributors in the biogas, but of course the major component remains methane. So because it is not including H2S, that is the reason why we have excluded this option. Then second one is saying methane, H2S and CO2, that is going to be correct. Then it says methane, H2S and O2. So this is incorrect, as the bacteria that are acting during the anaerobic sludge digester treatment, the name itself suggests they are anaerobic. So they cannot bear the presence of oxygen. So how they're going to produce it, right? So that is why it is also incorrect. And the fourth one is saying carbon dioxide and sulfur dioxide, that's also incorrect. Why? Because it's not including methane, which is the major component of it, right? So this is correct, the option number 2. So some part of the sludge could be used as inoculum in sewage treatment. Yes, this settling tank, right? So certain part will be used as an inoculum, right? This is an activated sludge that's going to be produced. Certain will be used as an inoculum back again to the aeration tank so that the aerobic bacteria can still perform the digestion of the organic matter, and rest all will go to the anaerobic sludge digester for further digestion part, right? So the correct answer is going to be option number 2, because there will be the formation of biogas, right? I hope this is clear to everyone. Now, moving ahead, another question that we have is the largest man-made ecosystem lacks all except, right? So when we talk about the man-made ecosystem, that means we are talking about anthropogenic ecosystems which are under the direct care of humans, right? So when we say about the anthropogenic ecosystems that's been created by humans or under the care of humans, so when we say that they have high diversity, that is not true. They lack high diversity, right? They'll be having, on the contrary, low diversity, because certain organisms can only be accommodated by the humans in that particular periphery. So that's why they have low diversity. Then it's say high productivity. That is correct, because of this extreme cared environment and the selection of these kind of organisms, there will be really high productivity over there. Then complex food chains, that's incorrect. In order to have complex food chains, you need a lot of diversity, right? A biodiversity should be really high in order to create these complex food chains. So that's also not possible because of the simplicity of these anthropogenic ecosystems. And then frequent circulation of nutrients, that's also not true, because this is not a self-sustainable system, right? The humans have to intervene during the formation and during the sustenance of this ecosystem. So there won't be a frequent circulation of nutrients over here by their own. So it will be going to lack all of these except the higher productivity, because that's being found in the anthropogenic ecosystems. So yes, you have correctly said it will be crop fields and aquarium would be the examples of it, right? So this is about the man-made ecosystem. Now moving ahead, we have another question which talks about identify the stage in which non-sister chromatids of chromosomes separate, right? So it is talking about the non-sister chromatids, right? So when we talk about these bivalents that we have discussed, so in this, if we name it like this, 1, 2, 3, 4, so 1 and 2 together will be sister chromatids, 3 and 4 together would be sister chromatids, 1 with 3 or 4 or 2 with 3 or 4 are non-sister chromatids. So they are saying that the stage in which non-sister chromatids separate, that means separation of homologous chromosomes, right? Another way of writing it would be the separation of non-sister chromatids. So separation of homologous chromosomes is a characteristic event of anaphase 1, right? Prophase 1, we could rule out this because over here crossing over and the synapsis is a key event. Here not the separation occurs, rather the conjoining or the what you could say association of the non-sister chromatids takes place. Also the exchange of genetic material between these. Telophase 2 could also be ruled out, because in telophase 2 already this separation has been occurred, and in telophase 2 disintegration of the what you could say not disintegration but decondensation of the chromatin material take place. Anaphase 2 would be the separation of sister chromatids, not the non-sister one, right? So this is also incorrect. So the correct answer would be option number 2, right? Moving ahead, we have another one, right? So this is all about the questions from this particular, chapters that we have took during this paper. And I hope this is clear to everyone. So keep practicing, keep focusing on your NCERT revision so that you can solve the questions with ease and try to understand all aspects that are associated with the topics. Thank you very much, everyone.